Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So today we are going to learn about Polycomb group of proteins and the Trithorax group of proteins. We are going to learn about how those two group of proteins they epigenetically regulate the sequential expression of certain genes which are quite important during the early development of Drosophila. So we will learn about the development of Drosophila in detail in the developmental biology, but let's quickly learn about the what are the life stages, what are the stages of life cycle of Drosophila. So I'm listing down the stages. The embryo of Drosophila. So the first embryo is formed. Then the next stage is larval stage, which turns into pupa, which turns into the adult. So this is the life cycle of the Drosophila. Okay, in the adult, gametogenesis happens, and then when gametes fuse, they form the embryo. Okay, so this is how the Drosophila is developed. So in the Drosophila embryo, so let's say this is the Drosophila embryo. So the segmentation of this embryo, it is decided by a certain group of genes, okay, and there is a sequential expression of those group of genes. So I'm not going to cover that in detail. So first, the maternal effect genes such as bicoid and nanos, they decide the anterior part and the posterior part. For example, the bicoid region marks the anterior part, while the nanos region marks the posterior part. Okay, so this maternal effect gene in turns activate the gap genes, and then there is sequential expression of pair rule genes, the segment polarity genes. Okay, and then lastly, homeobox genes, the homeotic genes. The expression of those homeotic genes, this is epigenetically regulated by the Polycomb group of proteins and the Trithorax group of proteins. So what happens is that it is decided whether certain genes will be activated at certain stage and certain genes will be repressed at certain state. So the maintenance of this activated or repressed state, that is maintained by Polycomb group of proteins or the Trithorax group of proteins. So Polycomb group of proteins, this maintains the repressed state of those genes, okay, while the trithorax group of proteins, this maintains the activated state of those genes. So for example, there's a gene, let's say there's a gene X, and this is activated. This needs to be activated during the development of Drosophila. Okay, so the active state of this gene will be maintained by trithorax group of proteins during the development, during the life cycle of the cell, and in the progeny of that cell. While there's a gene Y, and it is repressed. Okay, it needs to be repressed during the development. So the repressed state of this gene that will be maintained by Polycomb group of proteins. So mind me, Polycomb group of proteins does not do the repression of the genes. It maintains the repressed state. Okay, so this is not possible. So this is not causing the repression, but this is maintaining the repression. The trithorax group of proteins, this also maintains the active state of genes. Okay. In the development of Drosophila. So there's an interesting research paper. I'll post a DOI if you want in the comment section below. Okay, so this tells us this tells us about this interesting phenomena known as epigenetic memory. Okay, so let's learn about it. So what happens is that this is the Drosophila embryo, okay, and this is the anterior part and the posterior part. Okay, so in the homeotic genes there are two clusters. One is known as antennapedia cluster, okay, and other is known as bithorax cluster. Okay, so these are two clusters of genes which are important in the segmentation of Drosophila. Okay, so this particularly shows the bithorax group, bithorax cluster. The UBX, ABD1 and ABD2, abdominal 1A and abdominal B, sorry, A and B, all those three, they are consisted in the bithorax cluster of the homeotic genes. Okay, so this is the bithorax cluster of Hox genes. So this is the segmentation. These are the para segments of the embryo, Drosophila embryo. These are shown. Okay, and the color coding represents the certain genes. For example, the ABXBX, this gene is represented by faint orange, this one is represented by dark orange, and so on and so forth. So these are the enhancers of those genes. Okay, so these are the enhancer regions of those genes. So this is the enhancer region for the ABDB gene. This is the enhancer region for the ABDA gene, and so on and so forth. So these are the enhancer region. The color coding shows the this enhancer region. Okay, so orange means the enhancer region is for the BXT and PBX. Okay. So in the experiment, those enhancers of certain genes, like we have chosen this gene here. So the enhancer of that gene was coupled with the reporter gene, LAGZ gene here. So this reporter gene is going to report about the expression of the enhancer during the development of the embryo. Okay, so there are different cases here. Here there is only the gene enhancer. Here we have gene enhancer coupled with the Polycomb response element, while here the gene enhancer is coupled with the Polycomb response element of other gene. Okay, which is so this Polycomb response element. Let's say that there is a Polycomb response element which is present here, that is coupled with this enhancer. Okay, and here the enhancer is coupled with its own Polycomb response element. So these are the different cases. So what happens is that here it shows the early stage, early embryo, and here it is showing the late embryo. Okay, so what happens is that when the enhancer is coupled with the LAC, it shows that the half segment is showing the expression of the certain gene. So mind me, the bithorax cluster, the abdominal region genes, they are only expressed in this region. Okay, so this enhancer, this region, this is right. So they are only expressed in this region. But as the development continues, the expression of this gene is seen in overall embryo. Okay, when there is no Polycomb response element. But when there is Polycomb response element, their expression in the posterior region is maintained. Here you can see the expression in the posterior region is maintained. So this tells us that Polycomb response element, this is important for maintenance. Okay, for maintenance of the expression. Okay, so let me tell you why. Polycomb response element, PRE, Polycomb response element, they are the regulatory DNA sequences. And they do not have any they do not have a functional enhancer region, but those Polycomb response element are simply the binding sites for Polycomb group of proteins. Okay, so the Polycomb group of proteins, like the different complexes, PRC1 complex, PRC2 complex, the 4RC complex, all of them, they can bind to this DNA sequence, which is known as PRE. So the presence of PRE indeed signifies that due to the presence of PRE, now Polycomb group of proteins can bind to this sequence, and now they can maintain the repressed state. See, so the repression is done. So repression is already done. So this in this region the repression is already done, but it is not maintained in the further development because there is no Polycomb response element to which Polycomb group can attach. But here you can see when there is presence of Polycomb response element, the repressive state here is maintained. So that is why the expression of the enhancer is only in the posterior segments. Okay. So in this case, the PRE, which is coupled with the enhancer, that is from a different region. So this was done to check whether the PRE contains positional information or not. But the thing is, they found out that PRE do not contain any positional information. If PRE had the positional information, the result should have been something like that. Means the enhancer is going to express in this region, right? But if the PRE has the positional information, only this part will be repressed, and only this part will be expressed. So this PS12 will be expressed. So this green color region will be expressed because PRE is from that region. The rest of the part will be repressed. But this does not happen, and the expression is according to the enhancer. Okay, and the information about repression, it is the positional information, it is not contained in the Polycomb response element. This suggests us that Polycomb response element, it only maintains the memory. Okay, so this suggests that this Polycomb response element, the job of Polycomb response element is only to maintain the original state. So here it is the repressed state. So it only maintains the on or off state of certain gene which is already set in the early development. Okay, so whatever is set in the early development, that is maintained by Polycomb response element in the latter stage, but it does not have any positional information. Okay, so this is very important point, that PRE only maintains the on or off state. Means we can say that PRE, this has the epigenetic memory. So this plays a role in epigenetic memory. But the positional information, it is not contained by PRE, rather it is contained by enhancers. In this case, it is the embryonic enhancer, and this is the same in the larval enhancers also. In the larval stage also, during the early larva, whatever is set in during the early larvas, that will be maintained by the Polycomb group of proteins or Trithorax group of proteins. So that will be maintained. That memory will be retained by Polycomb response element, but the positional information will be contained by the larval enhancers. So this case is same for both embryonic and the larval enhancers. So the positional information is contained in the enhancers, not in the PREs. PREs only does the job of the epigenetic memory. So that's it for today. In next lecture, we will learn about the Polycomb group of proteins and Trithorax group of proteins, the main complexes that are involved in those group of proteins, and how do they maintain the active or the repressed state. Till then, see you. Bye bye.